Good afternoon, everybody. Today we'll be presenting to you our thesis project that is called after the title of Evaluation of Hypoglycemic Effects of Soursop leaves, Annona muricata, in methanolic extract. Our researchers team is formed after my classmate Emilio Alonso Venegas, Emmanuel Alejandro Téllez, and myself, Jorge Pimentel Fonseca. Our advisor is the Professor Carlos Rafael Manzano Palafox. Before we start to speak about the project itself, there are some concepts that are key to understand it, and we have to give you the definition. Starting with diabetes. Diabetes is a chronic metabolic disease that causes something we call hyperglycemia. Hyperglycemia refers that the normal glucose blood concentration are higher than the standards. There are different types of diabetes, but they share some characteristics such as the symptoms, which include the polyuria, the fatigue, the headaches, the polyphagia, and the sudden loss of weight. Among other affectations of diabetes, we found that this hyperglycemic state damage liver, kidneys, provokes also the well-known glaucoma, and has a very important damage in the vascular system. Among the treatments we have nowadays against this disease, we have the oral hypoglycemic and different types of insulin.
To continue, we need to define what is an hypoglycemic. These are substances capable of lowering blood glucose levels to normal levels that are found in healthy people. To achieve this, classic treatments are used, such as oral anti-diabetics and insulin in different representations. In contrast to this, new molecules are being researched, looking for effectiveness and efficiency based on new raw materials, such as vegetal ones. And that's where our object of study, Annona muricata, belongs. Over the past 2 decades, Annona muricata has been under investigation as an hypoglycemic agent and among its hundreds of properties. Annona muricata is a small upright evergreen tree that can reach between 3 and 9 meters in height. It is found throughout tropical America and the Caribbean. Among its principal properties, it is found to be cytotoxical, anti-leishmanial, antiviral, anti-cancer, and genotoxic. Also antimicrobial, and what concerns us more, its hypoglycemic activity. Annona muricata has an hypoglycemic effect due to the increase of production of insulin in the body, as well as an inhibitory potential against alpha amylase, alpha glucosidase, and lipase. These are enzymes that work by destroying the bonds found in polysaccharides, leading to a liberation of simpler sugars such as glucose and by consequence, increasing the glucose levels in blood.
Well, now we have understood this concept, we need to speak about the problem statement. We first need to take a look about the active cases. According to data from the World Health Organization, in the world, in the past year, there were more than 422 million of diabetes cases. In America, there are more than 62 million cases, and in our country, in Mexico, there are more than 10 million cases of diabetes. In addition to this, diabetes represents also a big expense for all the World Health Organizations. In the last year, more than 10% of the global budget of health was spent in diabetes treatment. This represents more than $700 billion. And well, speaking about these treatments, we find out that these treatments have a lot of disadvantages, such as the hard therapeutic adherence. This means that people cannot follow the treatment as it's supposed to be because it's very expensive or because it's hard to get it. Last but not least, we have to take a look about the quality of life. Quality of life is measured by an index, and diabetes is the disease that decreases most this index.
Okay, and to start giving shape to our project, we have to talk about what is the main objective of our project and what we're looking for with it. The general or main objective is the next: evaluate the effectiveness of the Soursop leaves and Annona muricata methanolic extract and as an hypoglycemic compound through in vivo experimentation.
And well, to understand the project, we have divided it in 3 stages. The first stages refers firstly to the acquisition and preparation of all the materials and equipment we'll be using during the project. In this stage, we will also be preparing the extract and some other substances we'll be using for the second stage. The second stage is itself the phase of experimentation in vivo with the rats. Here we'll be doing a lot of experimental process with the 24 Wistar Rats. And finally, the last stage is the statistical analysis and data collection. It will also include, sadly, but necessary, the euthanasia of the rats. And well, this will be the stage that will conclude our project.
Okay, we have to start to talk about the statistical analysis. Okay, we will do an statistical analysis to verify mathematically that we really verify our hypothesis in real numbers. For this statistical analysis, we will make a series of graphs in which we will measure the glucose reduction in the rats across the time. To evaluate these graphs, we will use a programming language called R, which tells us different significant values to be able to evaluate the glucose reduction, and we will see if our project was successful or not.
Here you can find the references that we used to make this presentation. And it's all from us. Thank you.